In this chapter, we'll be examining the process of sexual reproduction and meiosis. In the previous chapter, we examined mitosis, which is division of all the cells in our body, which are called somatic cells. There's one other type of cell, though, that's produced by our gonads, the ovaries and testes, which are called germline cells. So of all the cells in our body, the somatic cells, there's one other type of cells produced by the ovaries and testes that are called germline cells. These cells allow us to have a sexual life cycle and produce sperm and eggs. So the sexual life cycle is made up of meiosis followed by fertilization, which are all the somatic cells, and they have two sets of chromosomes. So every single cell in our body is diploid and has a homologous pair for each of the 23 different chromosomes. When we form gametes from the germline cells, they end up with only one set of chromosomes, and we call them haploid. Offspring will inherit genetic material from the two parents, so each parent contributes one chromosome to each of the homologs in order to form the zygote. Here we have a sperm with the 3 red chromosomes in his head, and then here in the egg we have 3 blue chromosomes. Upon fertilization, we form a zygote, which again is diploid and has 1 chromosome from each parent for each different homologous pair. So the sperm and egg are haploid, and they fuse to form a diploid zygote. Most life cycles have an alternation of generation. That is, we alternate between haploid and diploid stages. For us, we just explored the haploid sperm and egg stage, and they join together at fertilization to produce a diploid zygote, which will divide by mitosis trillions of times in order to form adult human beings. Some life cycles include a much longer diploid phase, in which there may be a haploid multicellular organism. Most of these organisms then will come together and give one cell to each other and produce a zygote and form a diploid generation also. We'll most often see that in plants. However, on occasion, we'll see some organisms that have a multicellular haploid stage and a unicellular diploid stage. That would be much like on campus. You had adult sperm and eggs of full size, multicellular size. They look different than each other, but each cell is only haploid. And then you had teeny tiny unicellular diploid people. But in most animals, the diploid state dominates. That is, the zygote first undergoes mitosis to produce many diploid cells. Later in the life cycle, some of these diploid cells, namely the germline cells, undergo meiosis to produce haploid gametes. Now, there are some special features of meiosis. On some levels, meiosis looks very much like mitosis, in that it's going to be composed of prophase, metaphase, anaphase, and telophase. We'll exclude the prometaphase, although each of these stages aren't really distinct moments. You have to keep in mind that everything flows, it's cyclic, so there is no actual defined prophase and metaphase. They flow through prophase, into metaphase, into anaphase. There's no snapshot place. But there are certain events that are described to happen in each of these phases, and they're very similar to what we see in mitosis. Meiosis includes 2 rounds of division, however. So we describe them as meiosis 1 and meiosis 2. Distinct from when we say mitosis. So when we speak of metaphase without saying metaphase 1, then we know that we're applying it to mitosis rather than meiosis. In meiosis, we'll always refer to metaphase 1 or metaphase 2. One of the key events that happens in meiosis that's different than in mitosis is the process of synapsis. This happens during meiosis 1, specifically in prophase 1, and it's when homologous chromosomes become very closely associated. As you can see here, there are 2 homologous chromosomes. One is blue and one is red. They become so closely associated that we see the formation of these synaptonemal complexes in between each homologue. Once they've become this tightly associated through the synaptonemal complexes, we call them a tetrad. So we have new terminology here. A tetrad is a homologous replicated pair during prophase 1. So the 4 sister chromatids together are a tetrad. Now let's remember, think about this. For the blue chromosome, which is composed of 2 sister chromatids or 2 molecules of DNA, the information on them is identical. On the red chromosome, we have information that is similar, but maybe a different version of the same information. So, for example, the blue chromosome might have blonde hair color on it. In the same location on the red chromosome, we do have an allele or genes for hair color. However, it may be a different version of that allele. It could be brown. It might also be blonde, or it could be red. So they have the same type of information, thus it's homologous information, but not identical. So we can term these 2 sister chromatids, the 2 blue ones, and the 2 red ones are sister chromatids. But what do we call this blue chromatid and this red chromatid? They are in the same homologous pair. We'll call them non-sister chromatids. So non-sister chromatids are chromatids that are in the same homologous pair, but are not attached at the centromere. They are attached, however, through the synaptonemal complexes. During this synapsis, as we'll call it, we'll see genetic recombination between those non-sister chromatids. So again, the non-sister chromatids are chromatids in the same tetrad or of the same homologous pair that contain different varieties of the same information. So a blue one and a red one. This genetic recombination or crossing over allows each homologue to exchange chromosomal material. We can see on the right-hand side here a diploid cell, and then the homologues have become replicated and they pair together. After meiosis 1, we'll see one of each homologue in each of the daughter cells. And after meiosis 2, we'll see that sister chromatids separate from each other into distinct cells, thus forming 4 haploid cells. So you can see the effect of crossing over here. If these 2 larger chromosomes, one red one from the mother and one blue one from the father, form a synaptonemal complex and have a crossing over event or synapsis, they'll change pieces of information such that one red chromosome has a blue piece in it, and the blue chromosome that went into the other cell, its homologue, has a red piece in it. They've exchanged some genetic information. That means alleles of genes that were formerly on separate homologues can now be found on the same homologue. So let's say, in this case, we exchanged the genes for hair color. We may see that this chromatid, the left chromatid, has blonde still. However, the right chromatid has this little blue piece, which actually could have been the gene for brown hair. So in this case, sister chromatids, the ones that are attached at the centromere, are no longer identical. Chiasmata is the actual site of crossing over. The contact between the chromosomes, as we'll see, is maintained until after metaphase, when we see the homologous chromosomes separate from each other. We'll see this in more detail coming up shortly. During metaphase, the paired homologs move to the metaphase plate and become oriented with the homologs of each pair attached to opposite poles of the spindle. When we were looking at mitosis, you would notice that the homologs operated separately. That is, they lined up single file down the center of the cell during metaphase. However, in meiosis, they line up as pairs on the metaphase plate. So as a tetrad, the homologs line up side by side along the metaphase plate. During anaphase, these homologs are pulled to the opposite poles. That is, the homologous pairs are separated. In mitosis, remember the sister chromatids were what were pulled to the opposite poles. And so in metaphase of mitosis, we separate sister chromatids, whereas in metaphase of meiosis, we separate homologous chromosomes, replicated homologous chromosomes, to be more specific. When we consider meiosis, the first division is termed the reduction division. That is because we're separating homologous chromosomes. We're reducing the chromosome number from 2n to 1n in meiosis 1. This results in daughter cells that contain one homolog from each chromosome pair, just as you see in the figure down here. They have one big chromosome and one smaller chromosome, one big one and one smaller one. The second meiotic division doesn't further reduce the chromosome number because, after all, each of these is a replicated chromosome. There are no more homologous pairs after meiosis 1 is complete. In meiosis 2, they line up single file down the center of the cell, just like in mitosis. So metaphase of mitosis and metaphase 1 in meiosis are distinctly different. However, metaphase 2 of meiosis looks very similar to metaphase of mitosis, because these are the two phases in which sister chromatids become separated from each other and pulled to opposite poles of the cell. So the reduction division is meiosis 1, because we're separating homologous pairs and reducing the chromosome number to half. After meiosis 1, in this case, we have 2 chromosomes in each cell. These ones are 2 replicated chromosomes. After meiosis 2, we also have 2 chromosomes in each cell, but because sister chromatids have separated, they are not the replicated version. So now we have 4 haploid cells after the completion of meiosis 2. Let's take a quick card quiz to review. Germline cells undergo meiosis to form genetic recombination occurs when homologous chromosomes contain the same genes but are inherited from different parents. Is this true or false? Which of the following is an example of a somatic cell? Here are your answers.